𝗤𝗂𝗽 from New Jersey, and I'm sure that the deliberation of Manoranjan 𝗤𝗂 will be very helpful for our participants. First of all, apart from this, I would also like to thank IES Institute or IES University for inviting me as chief guest of this function. I'm really thankful to Dr. 𝗤𝗲𝗄𝗏 for inviting me to participate and interact with such a high dignity participants. So regarding this webinar, I would be saying something about the good manufacturing practices, which is a quality management system. At a whole, if you see the good manufacturing practices in the pharma industry, it is a quality management system. And if you want to have drug with zero defect means there should not be any possibility of any defect in the product which is coming into the market. So the product should have zero defect, because if there is any defect, that will directly affect the patient, and you will be playing with the life of our patient. So one has to be very careful when he will go for manufacturing of any pharmaceutical product for the treatment of illness of the patient. So well, if you see the drug life cycle, then it starts from the research and development, where we develop some new formulations or new entity for the treatment of any disease, and for getting relief of the patient with that particular disease. So when we go for the research or development, our target is the human being, or say if it is veterinary product, then our target will be the animals. So if we developing any drug molecule or formulations for the human beings, then one has to be very careful. It's a very tedious process for developing a new drug molecules, but nowadays the trends are changed, right? We are taking the very potent old molecules and giving a new cloth, means new formulations, like making the target specific delivery system, so where that will deliver drug in effective concentration to the target site, and the element of any disease can be cured, and as well as the side effect of the drug, that potent drug can be reduced if we deliver effective concentration to the target site. So our intention, this is the research part, where we develop any formulation of any drug, whether it is any old molecule or any any new drug entity. Then after developing a formulation, it will shift to the industry level, means through the pilot plant. Then we see whether it is really feasible on the large scale manufacturing or not, because at our lab level, we prepare drug formulation at very low quantity, all right? So then you have to transfer your formulation, your research to the industrial level. That takes really a very, so many things we have to take into consideration when we transfer research from academic to the industrial level. Then same time, you will require the authorization from the appropriate authority, and then After, if the formulation is successful at the industrial level, then we manufacture it at industrial level, then we market it, and then after the marketing, you have to track the marketing issues of that developed drug formulation which is in the market. That includes the clinical trials, clinical trials, that is the clinical trial 4. This is a post surveillance. So we have to monitor all the time our drug product. This is a great responsibility of the industrial people to monitor. After making the drug formulation is easy, very easy, but marketing is very difficult, because it is not only selling the drug product, but at the same time you have to market it. You have to monitor it, whether it is giving the desired effect or not, or is there any side effect, and what is the trends of your formulation in the market, and how it is received by the patient and physician. So that is very important issues, and it is a great responsibility of the industrial people. So, well, if you want to bring a drug product with 0 defect, without any defect, then you have to work a lot. It's a responsibility of the pharmaceutical people who are working on the academic research institute as well as the pharmaceutical industries. So it's a great responsibility to pharmaceutical people or pharmaceutical scientists, right? So after that, if you want to make a quality product, then the 4 aspects, 4 steps we must have to take into the considerations. These are the good attitude, right? If you want to make a good quality product, or quality product, when I say quality product, it means it has no defects, right? So that good aptitude is very important in the making the good quality product, because if you do not have the good aptitude, then really you will not be able to produce quality product, right? Then the good material. Naturally, if you want to prepare a good quality product and having the good aptitude, but if you do not have the good material, then it will your product will not have the desired quality, right? So the good material is very important, and you have to then good system also. Good system is where you are making your drug product, right? So that the pharmaceutical industry, they should have very good system. And last is the good manufacturing practices. This is also very important aspect of the pharma manufacturing. The good manufacturing practices, all are come under the good manufacturing practices. If you have good manufacturing practices, naturally you will get good quality, because GMP is applied to your aptitude also, your material also, because at every stage you have to monitor your material, starting from the raw material which is coming into the pharma industry to the finished product which is coming out of the industry. So the input and output both should be from input to the output, both should be according to the good manufacturing practice. And same time, where the product is coming and from where it is going, you have to apply good manufacturing practices. So what is the birth of the GMP? How it comes into effect? That is very important. We must have to know that how it is come into an effect, that is starting from, if you see the history of the thalidomide tragedy in USA, you might be knowing about it. In 1962 in USA, it was a great tragedy of the thalidomide drug. It was using for the treatment of the sickness, right? And it was at that time it was prescribed for the pregnant women also, because it is said that it was said that this drug is safe for the pregnant women also, right? Or women who are bearing the child, for the child, then this drug is very safe. But after some time, when a report came, then the child, the women who had given the birth, the child was having the problem, the short of limbs, right? It was a great problem, right? And because this tragedy happened because of at that time the GMP was not so effective, and FDA people WHO was not worried about the quality product. Similarly, there are so many tragedy of the pharmaceutical product has come. Then there is sulfanilamide elixir also, where the alcohol was replaced with some something non-organic solvent, so it was having the toxicity, right? And because of that lapses in the manufacturing, not only by the scientists but at the floor of the manufacturing, it affects the life of so many peoples. That is why USD published in 1962, US FDA had published several new regulations, criteria for the good manufacturing practices, as well as the GMP to be followed in manufacturing, practicing, and holding of finished product. And in 1967, WHO has prepared a guideline for the GMP, good manufacturing practice, for manufacturing the pharmaceutical products. And it is the GMP is the part of the quality assurance aimed at assuring the products are consistently manufactured to a quality appropriate to their intended use, right? So this is the purpose of the GMP. So in 1967, WHO led a guideline, and they said that the product which you will be preparing, or pharmaceutical industry preparing, should have no margin for the error. That is, a product should have zero defect, and do the right time, and always we should follow the GMP guidelines all the time, starting from the input of the material to the output of the material, even though to the use of marketing of that material, that includes the distribution of the material. In 1967, Orange Guideline was compiled by the Department of Health and Social Services in UK. In 1988, GMP was adopted by Drug and Cosmetic Rules, legally adopted by Drug and Cosmetic Rules, and it is now called Schedule M. It comes under the Schedule M of Drug and Cosmetic Rules. So what is the GMP? That is a good manufacturing practice, is a part of quality assurance, which assures that products are consistently produced and controlled to the quality standard appropriate to their use. GMP is an integral part of the quality assurance. So quality assurance is the, under quality assurance, the quality control as well as good manufacturing practices come. Why GMP is important? If you see, it is a requirement, no doubt it is a requirement of the government. Why it is requirement of the government? Because they want the product which is coming to the market, it should not have any defect, so that patient should not adversely affected by some adulterated or the defective medicine which is in the market. Assure quality product, they have to assure the product which is coming into the market, it is the quality product. And as well as it, if you are following the GMP practices, no doubt it reduces the rejects and recalls of the products. Naturally, if the rejects and recalls of the product is reduced, the cost of the product is reduced, the burden, financial burden on the pharmaceutical industry can be reduced, and same time it satisfies customer also, right? And maintain manufacturing consistently, means that there will not be any variation from batch to batch. So the product which you are getting from the pharma industry, it is consistent. Company image, and it also affects the company image and reputation. That is why adaptation of good manufacturing practice is important. So the current good manufacturing practices, it is the quality management system, both current and good, right? It's and it applies to the to all aspects of the preparation, all aspects of the manufacturing of the drug. When a drug entity is intended for use of human being or target animal for animal drugs, right? In that case, you must have to adopt or use good manufacturing practices. Same time, this good manufacturing practices, these are the methods, facilities, controls used in the production of the drug, devices, food, and biologicals. These manufacturing practices are designed to ensure that, right? This is the very important, designed to ensure the safety, identity, strength, quality, and purity of the such product. So GMP practices ensure the safety of the drug, identity of that drug, strength, uniform strength of the quality and strength and purity of quality, strength, quality, and purity of the product, right? So question is then, what is quality management? Quality management, that is the related with the quality policy and intention and direction of overall the authority who are managing the pharma industry, right? So quality, it is an aspect of management function that determines and implement the quality policy. It is a duty of the management to impose the quality policy to the pharmaceuticals or to all those who are working in the pharma industry. The overall intention and direction regarding the quality as formally expressed and authorized by top management. So that also shows the intention and direction of the management. Then the quality management system, as I told you, it is the principle of quality management is to ensure the manufacturing products are fit for its intended use, complies with requirements, and does not place a consumer at risk. This is very important. It should not affect, we should not place consumers, means patient, at risk due to inadequate safety, quality, and efficacy measures. So to achieve these objectives, there are 4 quality measures we have to take. The quality assurance that meets the requirement of good manufacturing practices, good manufacturing practices. All manufacturing processes are clearly defined, systematically validated for the production of consistent quality product. I think regarding the validation, we have a very learned speaker, Manoranjan Sahu. He will tell you about why validation is equally important, and validation is the part of the quality management system, right? If you want to prepare a quality product, you want to prepare a product with minimum defect, the validation of all the process is of the pharmaceutical manufacturing is must. The quality control, it is also important. It is also part of the quality management. Quality control that focuses on sampling, specification, and testing the product before being released for sale or supply, right? So we have to frequently sample the drug product, and we have to see whether it is having the desired quality, desired specification, right, before the release of that product for the sale or supply. Then quality risk management. Same time, we must have to monitor the risk management system, a systematic process of assessing risk that can affect the quality of the product, right? So this is a very important aspect of we must have quality management system. If we do not have quality management system, then somewhere we are compromising with the safety, quality, and efficacy of the product. The GMP addresses several issues related to the productions, such as documentation and record. That is very important, because if we do not, documentation is just not keeping the record aside, just putting the value and keeping the record aside. We must, same time, we must have to analyze the data which you are getting during the manufacturing of that product, right? We must have to analyze, and accordingly we have to modify in our different SOPs, standard operation procedures, right? So that our quality of our product can be improved. Then it implies to process and production control, physical plant, laboratory, and facility design that is related with the premises where the product is manufactured, and personal quality control oversight. Personals means the workers who are working there for the production of the pharma product. So that is in personals also, which we must have to apply GMP process guidelines. Maintain and calibrate equipment, that is also very important. We have to calibrate equipments or machines which we are using for production of the product, because whether they are giving the desired output or not, we must have to calibrate it and we must validate it. Clinical trial required. If the clinical trials are required, it should be according to GMP processes. Then it also addresses with the material management, input and output material, packaging and labeling of the pharmaceutical product, storage and distribution, in every aspect of the pharmaceutical processes. The GMP is applied. We must have to follow the GMP guidelines for getting the quality product, efficacious product, safe product. Laboratory controls, rejection and reuse of the materials, complaints, recalls, agent, broker, distribution, repackers, labelers. We must have to apply GMP regulations. So these four are intermezzos, 5 intermezzos that play a very important role as far as the GMP regulation is concerned. The premises, the primary materials, pupils who are working in the pharma industry, procedures which we are following for preparing the product, process defined and recorded. All these 5 P's are very important. Then basic requirement of good manufacturing practices, clearly defined systematically review process. This is the requirement of the GMP. Qualification and validation is to be performed. It is the validation is the part of the GMP, right? But if you are going for the validation, then the qualifications, we must have to define We must have to specify the qualification of that particular machine or that process. We must have to, if we don't know what we desire, right? Means what type of quality we need, then validation is having no use. Appropriate resources are provided, like qualified and trained personnels, right? Premises, space, equipment and services, materials, container, labels, procedures, storage, transport, laboratories and in-process control, as well as clear written instruction and procedure is SOPs. We must have SOPs for all the processes, all the machines, trained operators, that is related to the human resources who are working in the pharma industry, and record of action, deviation and investigation, record for manufacture and distribution, proper storage and distribution of the raw material as well as the finished product, system for complaint and recalls, all are the requirement of the GMPs. So it is a continuous urge for the improvement. GMP is the continuous urge. It's a continuous process. It doesn't mean that at this moment you have frame a guideline. All right, you do not have any now chance for improvement. Always improvement is there, and so that is why it is a continuous urge for improvement. It's a dynamic process. If we need some improvement, we must have to do it. Involvement of the management. How will you see that improvement is required? By involvement. If the management is really involved in all the process, if he is observing all the process, only then he can see the improvement. Annual product quality review. We must, if the quality of the product is reviewed annually or biannually, then we can have some improvement in the process, in the product quality. Complaints handling. How you are handling the complaints. If the complaints are coming from the market means from the patient, then how you are taking it? You are not taking it personally. He is complained that, and you are just neglecting it. And if you are taking it seriously and you are analyzing it properly, and then you are modifying it in your process, right? Then you will get, your quality will certainly improve. Self-inspections, right? So these are the different urges for continuous improvement. Then what is the production goal, right, of the pharma industry? The safety. Safety is that is our top priority. Safety of our workers. So product is free of unwanted side effect when used appropriately by patient, right? Then identity. Product exactly matches the labeling and related documents. Strength. Product has strength. Strength is really directly connected with the therapeutic entity which is present in the formulations or dosage form. The product has correct concentration, potency, or therapeutic activity of the active ingredients. Only then you will get the intended, the response of that product. The purity. Purity product is free from any contamination. Single 0.0001% contamination may your contaminations lead to may affect the life of the patient. So there should not be any contamination, and we must have to review it properly. Quality product meets all the expected qualification, performs as claimed, right? Product made consistently, safe, effective, right? Then I will test a little bit about the process validation 2000 guidelines 2011. FDA current thinking evolves on process validation around process design, process qualification, and continuous process validation. I think Mr. Manoranjan will take all these aspects and elaborate on these different stages of the process validation. So simple process design, that is related with the development and scale-up activities. Then process qualification, that is demonstrate reproducible manufacturing through the conformance lots. Then routine continue process verification, that is a routine manufacturing monitoring of the performance. It's a continuous process, or in process, we can say in process monitoring of the product. Then validation and qualification. As in the starting, I told you that if you want to validate, then we must have to know the qualification of that particular process. Only then we can validate. So it is a qualify system, premises, equipment, if they are fit or ready for its intended use, and validate if process and procedure can repeatedly produce high quality product. Critical step. It's a critical step in the manufacturing process, and should be verified to ensure that product quality is consistent and maintained at a high level. So according to WHO, qualification and validation should be established and provide documentation stating that the design qualification, that is related with the premises, supporting utilities, equipment and process, have been designed in accordance with the requirement for the GMP specification. Installation qualification, all these related with the built and installed in compliance with their design. If it is properly built, premises according to qualification, if it is built, then it is as per the design, then it comes under the installation qualifications. And equipment is properly built as per the design, then installed properly as per the design, then it will be installation qualification. Operation qualification, the premises supporting utilities and equipments operates in accordance with the design specification. That will come under the operation qualification. And performance qualification is, this is the specific process will consistently produce a product meeting its predetermined specification and quality attributes. This is the GMP right, good manufacturing practice right. In this case, GMP is applied to all the process of the pharma industry, starting from the personnel, building, warehouse. Warehouse means where you are storing the raw material and finished product. Then machine and equipments, production, filling and packaging, documentation, and quality assurance. I will touch a little bit about the documentation and quality assurance, because if we touch all these GMP requirements of personnel, building, warehouse, machine, equipment, production, and filling, that will take a lot of time. But in short, I will tell you about these GMP requirements for the personnel, building, or warehouse, machine, equipment, production, and filling, and something more I will elaborate about the documentation and quality assurance. So, well, this personal first is personal, means the workers who are some authorities who are working in the pharma industry. So there should be list of government authorities. We should have, the pharma industry should have the list of government authority to which we are answerable, and who is responsible for that regulatory body. That we should be aware about the those authorities. And organizational chart, every pharma industry should have organizers chart, like the key personnel and their duties and deputies. In organizational chart, where is quality assurance and whether there is a separate person for quality assurance or GMP related jobs, job description, responsibility authority, and we must have to fix it to the key personnels for describing the job and responsibility for the personnels who are working in the pharma industry. Then the appointment, it is related, starting from the appointment of the personnel to the health precautions of the personnel who are working. So every aspect of the workers or authority who are working in the pharma industry should be according to GMP guidelines, means appointment of the personnel. It should be as per the requirement of the pharma industry, right? Whether the personnel is trained or not. If he is not trained, then you have to train them. Then training of the personnel, how you are taking the giving the training to the personnel or appointed persons who are working in, who will be working in the pharmaceutical industry. The entry procedure, what will be the entry procedure for the entry into the pharma industry or different departments of the pharmaceutical industry, working hours and breaks. All will have the standard operating procedure for appointment of the personnel, training of the personnel, entry procedure, working hours and breaks, and clothing procedure. If he has to change the cloth, then there should be a standard operating procedure for clothing procedure, right? There should be separate room for changing the cloths, right? It should have, say, air curtain and should have proper lighting system. All these things comes under the clothing procedure. Personal hygiene, that is also very important, and health precautions. These are the two aspects that pharmaceutical industry must have to take care of the workers who are working in the pharma industry. Regarding the personnel, their hygienes and health precautions, right? If say if we take an example, if a person is having some cuts in the hand, right, then he should not allow in the production area or the area where his cuts will come in direct contact with the product. So these are the different. There are different guidelines, GMP guidelines for all these steps. Then building and building technology. GMP guidelines are there for the building where the pharmaceutical product is manufactured. So according to that GMP guidelines, there should be a building plan and what will be the feature of the building that should be defined properly. And we have some map of the building and of the electric connections, of the water, and of entry point. Everything is defined in that plan, building plan. The material flow, that is also very important. So when we plan to construct any building, then we must have to take care of the material flow. There should not be cross contamination of the material, right? The human beings should be moved from a different corridor, and material should come from different corridor. There should not be direct contact of the material with the workers who are working in that area. Plant room surfaces, you must have to clean it properly. These are the figures. The surfaces should be smooth. There should not be any cracks in the wall, right? And the wall and ceiling where is wall of flow, where it is meeting, it is covered and it should have curved, right? It should not have sharp edge, so that if it is having the sharp edge, then it will be very difficult to clean. So this all during the designing or during the construction, we must have to take proper planning and its implementation. And lighting is very important. Say every corner of the manufacturing area should have proper light. there should not be any shade of the person over the product which is manufacturing. So we should have proper light. We should avoid the sunlight, because sometimes sunlight may quality of the product. So we should avoid the direct exposure of the product with the sunlight. Then waste from the building water supply. Then water supply is also very important. How you are, which water you are using, demineralized water or water for injection you are using for making the product. So this is very important, water supply, how the water, frequent testing of the water for the chloride, sulfate, and TVC is very important. The water which we are using for the preparation or for the manufacturing of the product and cleaning of the your containers, that water quality should be ascertained. And social facility and changing room. Every pharma industry should have some recreation centers according to GMP guidelines. Preventive maintenance for building means if you see this picture, all right, then you will see that it should be properly green. Outside the factory should be properly green, and there should not be any dust or what we say leaves, dry leaves on the open space, and the surface should be asphalted, properly asphalted, so we can avoid the dust. Then cleaning and disinfection of the all the floors and machines is very important, and the air distribution handling system. Nowadays, it is the mandatory. Every pharma industry should have a AHU, air handling unit, right? And it should be properly installed and properly monitored for the clean air which is coming and which is coming out of coming in the production or manufacturing area and coming out of the pharma industry. I think it is, yeah. So, well, this is the plan example of the material and people flow in the pharma industry. So there should not be any cross contamination of the material as well as the workers who are working in the pharma industry. The red one is the clean zone, where it should be cleaned properly. That is weighing, processing, washing, filling, that come under the red zone. Under the green zone, the packaging is taking place. And the blue zone, that is the control one, where the say incoming goods, from here incoming goods is coming and it is stored, raw material and packaging material stored here, and from here it is issues for the processing, right? This is the corridor for the movement of the people, and from here outside the utilities and services are giving from the backside of the that particular department. So there should not be any contamination of the workers who are working with the material. Then warehouse and dispatch. Under this, the warehouse means where your product is stored. The raw material, it is starting from the raw material to the finished product. Raw material means raw material as well as the packing material that which is coming into the premises. It should be properly screened for the say that is the under the we must have to keep label, and we have to check it whether it is according to the purchase order or not, And then we have to take, after receiving the product, we have to take sample, then we have to send sample of the material, whether it is raw material or testing material, for the testing for its quality. Then we'll distribute on the basis of first in, first out in the store. And we must have to measure the safety. We have to take all the safety measures during the storage of the material. We have to segregate material, right, which are by different levels. That is the yellow, green, and red. Yellow, green, and red. So this material under test will be yellow. We put yellow label. Approved is green label, and rejected if the material which is coming into the which is in the store is not having the appropriate quality or quality which we desire, then we put the label of rejected, and rejected material should be keep away from the all these under test or approved materials. Generally, the sample container have the name of the material. After taking the sample from the material, right, there should be separate sampling room where we take the sample from the container. We have to weigh carefully the sample, the material, as well as the person who is taking the sample should not be contaminated. And on the sample, we put the label, name of the material sampled, lot number, container sampled, and sample date and sample collected. Name of the person who is collecting the sample. Then machine and this equipments, and the machines which are and equipments which are present in the pharma industry, whether the machines are installed as per the design and performance. So installation, design and performance, operating instruction, clean distribution, calibration, and validation. All these are very important ones. Then production, that is related with the production of the particular dosage form, the formulations, right? We must have a written instruction like manufacturing formula code, batch manufacturing record. All these should be, during the manufacturing, we must have to follow all these MFC and VMR, batch manufacturing record. So according to that, we must have to prepare our formulations. Then prevention and contamination, we have to take, there are all having the SOP, how we will process the material in the manufacturing area. So to prevent the contamination of the product in process control and during the processing, during manufacturing, quality control persons will come and take the sample and see the product which is manufactured in the manufacturing area is having the desired quality or not. So frequently we have to take sample, batch coding. The coding, there are SOPs for batch coding, product related information, and reprocessing. If there is a reprocessing, there should be SOP for reprocessing of the different formulations. Then we have to validate all the machines, persons who are working the area. We have to validate properly. Semi-finished and bulk product should be stored separately. They are having, all these are having the GMP guidelines. Then filling and packaging area. This is during, after manufacturing, then the material will shifted from the manufacturing area to the filling and packaging area. There are written instruction SOP for filling and packaging of a particular product. Preparatory operations, accidental contamination. We must have to avoid contamination during the preparation also. Then coding, yielding, in-process control, product process related instruction, and finished product, right? So they are having the GMP guidelines. All the steps are having the GMP guidelines, SOPs. According to that, you have to manufacture that product or pack that product. Then Then documentations. Different documents you have to prepare during the processing of the pharmaceutical products. That is very important during or the product for the manufacturing of the product. We must have to use permanent ink like black or blue ink, right? For writing the documentation, it should be eligible, clear, concise, accurate, timely prepared, and consistent, and should be complete or in all aspects. The legal requirements of the documents, if you see that they are comprehensive in content. All the documents should be a comprehensive information of the manufacturing. So it should have the product specification, manufacturing specification, packaging specification, master formula card, all right, batch manufacturing record, batch packaging record, SOP for every important activities, control specification, control record, distribution record, all should be, all these records we should have. This is the requirement of the legal requirement, and the specification of the documents. If you say our specification are available for every starting raw and packing material, which include organoleptic characteristics also, right? Our specification available for auxiliary materials like disinfectant material, fumigation material, pesticide, lubricants, which may have indirect effect on the product quality. Is it assured that no change in the specification is done without approval of the quality assurance people, R&D or production, even for the purchase, economy purchase? Is there periodic review of specifications that we must have to ascertain? And the master formula, that master formulation code, is the master formula available for each product and batch size? The ideal content of master formula is name, strength of the product, generic brand name, brief, chemistry and category of active ingredients, production number, batch size, effective data review date, name, 𝗵𝗵𝗿𝗆 status, weight of each ingredient, total weight of doses form. All right, these are the content of the master formula code. Specifications of the material, sieve size, witness, all these must have to mention each and every step of that production of that particular formulation should be mentioned in the MFC, right? So these are the different specifications that we must have to put in the MFC card, right? And this will be the card will be used by the person who is involved in the manufacturing of that particular formulations. So these are the different aspects, and then batch manufacturing record. This is also very important. That traceability is very important. ऐसा नहीं कि जब batch manufacture हो रहा है तो record ढूंढे जा रहा है कि कहाँ है वो record. So it should be easily traceable, should be placed in such a place where it is easily traceable. Date, time of every step, dispensing record, preserving the dispense level, person doing and supervising the step, line clearance record, manufacturing expiry date, in process quality control checks. BMR should always travel with the batch process, right? So whenever that batch is coming into production, then the BMR, the batch manufacturing record, should come along with that production of that product. Review of the BMR by quality assurance before release, investigation of a discrepancy in BMR by quality assurance of production, procedure for investigation to batches before and after the concerned people. Then distribution record, then record for log books, all this should be maintained. Returns, if some goods is returned, then we must have record and SOP for the returns also, right? And how will you handle the returns? Then complaints, how will you handle the complaints? We must have record of the complaints and how you are, what is the SOP of the handling of the complaints, right? And how you are handling the complaints. Then production recall procedure. If the product is rejected, then how will you recall the product from the market? So there should be SOP for product recall due to internal observation or FDA enforcement. Easily available for distribution records, reprocessing and distribution of recall product, that should have proper procedure. Last one is the quality assurance. The quality assurance is the organization chart of quality assurance. Is any analysis quality means we have to assure the quality of the product. There are now a section. When I was in the pharma industry in 1984-85, long time back, 30 years back, 27, 28 years back, and then there were no concept of quality assurance. We were having only the quality control, right? Now, and GMP process was there at that time, but there were no quality assurance. Only quality control people working as a quality assurance people. He has to ensure the quality, assure the quality of the product. So is any analysis carried out by the outside party regarding the quality of the product is concerned? Consent letter from outside party. If it is from the outside party, then we must have the consent letter. So you have to maintain the record of the quality assurance. Audit of outside facility of its qualifications is important. Abhi to dekho, if you see the pharma industry, every step audit hota hai. Every step is auditable, right? Because it is a responsibility of pharmaceutical industry to assure the quality of the product. Authority, responsibility of the quality assurance, checklist of the batch manufacturing record, all are SOPs approved by the quality assurance or production. ऐसा नहीं है कि प्रोडक्शन वाले ने बना लिया रिकॉर्ड, SOP बना ली, कोई भी प्रोसीजर बना ली, या मैनेजमेंट who are person who are sitting on the top management उन्होंने एक प्रोसीजर आपको दे दी, इम्पोज कर दिए. नहीं, it should be prepared by the qualified person, right? Or qualified who are having a good experience of quality assurance of production. So that should be approved by the SOPs. Question बनता है कि whether manufacturing SOPs can be changed. Sometimes, generally we do not change the SOP which is approved by the quality, but if the quality of the product is compromising, in that case we have to change it. आपको एक मैं छोटा सा experience share करना चाहूँगा. When I was in Franco-Indian Pharmaceutical company, the Bombay, they were manufacturing 𝗏𝗏𝗃. And in the manufacturing of 𝗏𝗏𝗃, they were using the hemoglobin. And hemoglobin is coming from the slaughterhouse. So being the Jain, I'm the Jain, being the Jain, if you want to test the quality of the hemoglobin which is coming from the slaughterhouse, then you have to take a spoonful hemoglobin, teaspoonful hemoglobin from the container, and you have to test it. Whether it is having the proper quality or not, means वो contaminated तो नहीं हुआ, false तो नहीं हुआ है, उसके degrade तो नहीं हो गया है. So that is very important test. So whenever these gains of the hemoglobin coming into the production area, the production chemist has to, or quality control person has to check all the gains. The hemoglobin which is coming into the production area or hemoglobin which is being used in the production of 𝗏𝗏𝗃 is having the proper quality or not. So this is very important one. Being the Jain, I also supposed to take a teaspoonful hemoglobin, but हमारे religions में we cannot take any material उनका जो ओरिजिनेटेड फ्रॉम द एनिमल्स, एनिमल जो होते हैं ना उनका हम लोग इस नॉन वेज नहीं जनरली लेते हैं। तो मैंने कहा था आई विल नॉट टेस्ट इट। मेरी नौकरी लगी हुई थी, मेरे को वहां पर अभी 2 महीने हुए थे, एंड आई स्ट्रेट फॉरवर्ड सेड दैट सर आई विल नॉट टेस्ट इट। मैं यहाँ काम कर रहा हूँ, दैट इज ओके, अगर इफ यू से देन आई कैन क्विट। तो production manager took me to the managing director, and he said that Mr. Jain is saying that he will not test hemoglobin, then how we will run this production? It is very difficult one. फिर क्या हुआ कि it becomes an issue. मैंने कहा सर ऐसा है कि, and he said अच्छा ओके नहीं, किसी दूसरे को, it is written in the SOP, you have to test it. हम किस SOP में कोई भी चीज, if anything is written, we cannot avoid it. We have to follow that procedure. अभी जब SOP में लिखा है कि आपको टेस्ट करना है, you have to test it. अभी तो मैंने कहा कि सर आपको अपना SOP, you have to change your SOP, आपको चेंज करना पड़ेगा SOP। कहना नहीं SOP चेंज नहीं होता है। तो हमने कहा अगर SOP चेंज नहीं होता तो नहीं करेंगे, हम आपको जो प्रोसीजर बताएंगे that procedure is more reliable as compared to the procedure which is written in the SOP for testing the quality of the hemoglobin। मैंने उनको बोला कि ये आप instead of testing it, आप अगर उसको स्मेल करेंगे तो आपको पता लग जाएगा हीमोग्लोबिन की क्वालिटी ठीक है कि नहीं। You will not believe कि after that a test was conducted कि एक chemist है who was expert in analyzing this quality of the hemoglobin, और उसके सामने मेरे को खड़ा करिए। There were 25 cans of 40 liters each। कैंस आता है स्लॉटर हाउस से, वहां से कैंस आता है हीमोग्लोबिन का। अब ये क्वेश्चन इज, तो मैंने उनसे बोला कि बिफोर ओपनिंग द लिड ऑफ दैट कैन जस्ट अलाउ मी टू टेस्ट इट, देन आफ्टर दैट द केमिस्ट another chemist will test it। फिर उसके बाद उन्होंने क्या किया कि उन्होंने कहा ओके आप जैन साहब करेंगे इसको टेस्ट कैसे करते हैं। हमने सारे लिड को खोला एंड स्मेल किया, एंड आई फाउंड 3 cans 3 cans were having the inferior quality hemoglobin, मतलब hemoglobin was deteriorated, वो खराब हो गया था, फॉल स्मेल आ रही थी उसमें। फिर उसके बाद जो टेस्ट हुआ कि केमिस्ट जो पर्सन था उसने देखा, तो उसने बोला कि सर जैन साहब ने जो 3 कैन बताए हुए हैं उसमें 2 कैन है जो खराब हो गए हैं, 1 कैन खराब नहीं है। अभी क्वेश्चन बनता है कि जो 1 कैन जिसको मैं खराब बोल रहा हूँ, वो केमिस्ट जो 1 टीस्पून फुल हीमोग्लोबिन लेके टेस्ट करके कर रहा है, वो ठीक है कि जैन साहब ठीक है। आफ्टर दैट जो कैन था 3rd कैन, उसको माइक्रोबायोलॉजिकली टेस्ट किया गया, एंड दे फाउंड दैट वो कैन का हीमोग्लोबिन खराब था। यू विल नॉट बिलीव, उसके बाद SOP चेंज हुआ, और जो मैंने प्रोसीजर बताया उसके बाद वो अडॉप्ट हुआ, वो उसमें इंकॉर्पोरेट हुआ। तो it is not necessary sometimes what happened, but it is really a tedious process. Once the person should be assured that if you want something change in the SOP, and if it is not compromising the quality of the product, or it is improving the quality of product, then certainly they adopt the changes in the SOP. So decision generally taken by the quality assurance and production man, audit of contract manufacturing by quality assurance in case of the contract manufacturing, audit of vendor by quality assurance. Vendor means who are supplying the raw material and packaging material, उनका भी ऑडिट ये लोग फार्मा इंडस्ट्री वाले करते हैं। Then equipment and facilities in quality assurance, all this there should be list of the quality control equipments, ventilation, physical testing, chemical testing, microbial testing, packaging material, reference sample storage, stability testing. There are so many aspects that we have to consider. It's a great responsibility of quality assurance. हम तो प्रोडक्शन में केवल हम प्रोडक्शन करते हैं, क्वालिटी कंट्रोल वाला सिर्फ टेस्ट करता है. जो quality assurance person is between the quality control and the manufacturing, or as well as the finished product distance, उसकी responsibility बहुत ज़्यादा होती है quality assurance person. And one, we have to see the quality person, quality assurance person who's working on the pharma industry. He should be qualified, properly qualified, should have proper experience. Only then he can perform his duty properly. So in laboratory controls, there are SOPs for volumetrics. कभी-कभी होता है आप volumetric flask में, even our lab also, हम 100 ml का volumetric flask लेते हैं और फिर हम next day फिर दूसरा volumetric flask ले लेते हैं, तो हमारी वही reading, वही sample है, वो change हो जाती है, right? It means the volumetric flask which you are taking in the laboratory, that is not calibrated. So all the measuring equipment should be properly calibrated, or the machine should be properly calibrated, or the equipment should be properly calibrated. Only then you will get consistent result. Testing material, these are all responsibility of quality assurance. Then release and of distribution. Right, during the testing, each batch and random batches of non-parental tested for bioload, right? It is tested for bioload, biological content. Is there SOP for sampling finished product indicating number of units to be sampled per container? कितने sample लेना है? There should be a SOP. Is every analytical method validated? SOP for handling of batch that fails in the testing? And who signed the release document? So if the material is approved by the quality control, then it will come to the approved and it will be released from the premises. And right, if it is rejected, then we put a label rejected, then necessary decision will be taken on the rejected finished product. So these are the documents to be maintained in the quality control, right? Then stability testing, there should be a scheduled program for the stability testing of the, right? So these are the different guidelines, GMP guidelines for stability testing, microbial testing, guideline for microbial testing. Then these are all the guidelines for the different processes which are taking in the pharma industry, GMP guidelines. And if you want a quality product, then we must have to follow the current good manufacturing practices. Only then we can get the quality product with zero effect. If there is a violation by the GMP, then severe consequences will be taking place, like product is adulterated, product may be defective, shutdown of manufacturing facilities. If it is found adulterated or defective, there are chances of shutdown of the factory or pharma industry, the seizure of the products, then recall of the product. You have to recall the product. It is a costly affair. Front page news. If some factory is shut down, or if your product is having some defect, right, or impurity, and you have to recall the product, then it becomes a front page press coverage and competitive disadvantage. Market, in the competitive market, it is a disadvantage. It affects the marketing of your product, different products. So GMP holds on product applications. Injection and consent decree also is also be chargeable. Say sharing a blog. Once upon a time, it was charged $500 million. Abbott Laboratories $100 million. 𝗦𝗃 Laboratories, $30 million, individual defendants. So there are, if you are compromising with the guidelines of the good manufacturing practice, it is not good for the pharma industry as well as the consumer of that product. Criminal investigation and indictment, this is, and lawsuit. These are the consequences, severe consequences of the GMP. So in short, in summary, we can say pharmaceutical industry is regulated by good manufacturing practices. Good manufacturing practice must be followed. It should be seriously followed, right? For getting the product with zero defect. GMP ensure drug products are safe, pure, and effective. So if we do not do, then who will do? Who would do it? This is important. We have to do. We have to be qualified. Thank you very much. Over to Harpreet.
Thank you, sir, for sharing all the basics of GMP and quality assurance in precise, easy, and brief manner. We really appreciate you taking out time for us from your busy schedule. It's always been our pleasure to have you with us. Thank you for gracing us with this fruitful information from your experiences. Now, moving forward, I would like to request our guest keynote speaker of this webinar, Mr. Manoranjan Sahu sir, to kindly address the audience.
Before switching over to the say Manoranjan, I would like to say that can I leave this meeting? Or if there is any question, we can take 2-3 questions. Then even Manoranjan can help us in answering if I am not able to. Harpreet.
Yes, sir.
Sure, sir. If you have got some work or if you are getting late, so we will ask the questions to Sahu sir. There is no problem, sir.
So no, it will not be any problem. So can I leave because I have another meeting at say 7 o'clock.
Okay.
Still
okay.
have time.
Oh. Okay, sir. Thank you very much, sir.
Thank you, sir.
Thank you. Thank you. Thank you all. Thank you all. And can you connect Manoranjan for a while so I can interact for say before starting his deliberation or his lecture? Manoranjan, is it audible to you?
Yeah, I'm hearing you clear and clear. It was nice meeting you. Actually,
same here. So well, you are in New Jersey.
Yes, right now, yes.
Yeah. From where you did 𝗵𝗵𝗿𝗵?
Well, I have done my pharmaceutical both bachelor and masters from the Simanta Institute of Pharmaceutical Science in Odisha. So it comes under the Biju Patnaik
Simanta,
University.
I know Simanta, yes.
Yeah. So I have done both the bachelors and masters from there, and then after 12 years in working in India, I came here to Japan and then USA. So I'm working right now as an investigator. So that's all a brief history of mine.
So still you are in pharma industry, yeah? You can very good. Can you share your details with me? So in future, if I need your help, I can contact you.
Yeah, I can. I will just if you can give me your number.
Yeah.
I will note it down
please.
and I will forward you a message to you. So can you give me a mobile number if it is possible?
94251
94251
72
184.
184.
Okay, I'll drop you my contact details on that.
And email also. You can write on my email ID also.
Okay.
It is Dr. Short form of Dr. �11SK Jain, Jain, �11SK in. After Jain, put in, all in small letters, @gmail.com.
Okay.
Same is on yahoo.com also.
Okay.
Right?
You're right.
So Manoranjan, I will interact with you. Right now, as you know, I'm heading the Department of Pharmaceutical Sciences. You might have heard the name of the Sagar University. And
Yes,
our
it's a very
Department of
famous
Pharmaceutical
university.
Sciences is one of the oldest department of the country.
Yes,
we,
absolutely, I agree to that.
2016, we completed 60 years. So if you go anywhere in the world, you will find alumni of the Sagar University. Okay.
Definitely.
So keep in contact, right?
Definitely.
And I will require if you require any help, I will contact you. And if you need any help from our side also, right?
Definitely.
Always available.
Definitely.
Our department is always available. Okay.
Yes.
Thank you very much. Thank you.
Thank you.
Yeah, please. I will see. It was just a few minutes. I will be with you. Our meeting still. I am having the 10 to 15 minutes. So
that will be very graceful of you.
Yeah, please. You continue with your lecture. Yes.
Okay.
Harpreet, now.
Yes, sir. Now, I would like to request our guest keynote speaker of this webinar, Mr. Manoranjan Sahu, sir, to kindly address the audience.
Okay. First, everybody can see my screen right now.
Yes.
Yes,
yes,
sir.
And since this will be an webinar, and the best part of a webinar is we don't need to ask, okay, can you hear me at the back end of the branch? So moving forward, my lecture is about validations. What I will request all the students who are on the line is that they can stop me in between and ask questions pertaining to that particular slides or any questions that they have, so that we can go on addressing the issues as we see them on these slides. So please feel free to come up with your questions as and when required. I'll be very happy to answer as I go on. So as we have already seen in the GMP slides, there is one particular slide which I was very impressed, was the attitude one. Absolutely spot on is that in the industry, when you will join, after you're completing your degree, one of the key aspects that I look forward while hiring any new person is the attitude. Without the correct attitude and without a correct learning spirit, it doesn't suit in the pharmaceutical industry. Though it is not written anywhere in the guideline, you will not find those written in many of the textbooks. But the right attitude is one of the key factors for developing a very good quality system in any organization, and that's what we emphasize as a part of the quality culture at Lupin. And also we emphasize the integrity of a person very highly in our organization for which I work. So moving on to the note, as we have already shown in the previous slide, which Mr. Jain also shared, is that yes, there was a 211 guideline which laid down the principle of process validation from the FDA. After that, European Union in 2015 came out with a guideline which was implemented in October 2015. So all this guidelines came, and then we started to look at the process monitoring in a very different way. Now, by basic definition, if you ask, and like any questions that you will get in any interview, what is the definition of a process validation? Come on. This is what everybody gives me. It's a collection and evaluation of data from the process design strategy through commercial production, which establish scientific evidence that the process is capable of consistently delivering a quality product. Now, the key definition that we all remember this definition like a mantra, but the key aspect of this mantra is that we must understand each and every word that is there in the definition. First is the collection. When I say collection, as you will go through the slides, you will find it consists about how the collection is done, what is the sampling methodology, from where the sample will be collected, and why the sample will be collected are a part of that collection. So it's not just a word, it's actually an entire chapter that is defined in a single word called collection. And evaluation of data, as also Mr. Jain said in between, if somebody has listened to the slides correctly, the evaluation of data is as important as collecting the data. What is the value of information if we are not able to evaluate it? Now, if I know there are 2 cats in my house, okay, I have 2 cats, but if I don't know the name, if I don't know what they are doing and how old they are, I don't make any sense of having a 𝗂𝘃 in the house. That's a very generic and a very simplified. So evaluation of data means the statistical analysis. Do you do a Pareto analysis? Do you do a control chart? Do you do a P chart analysis? Or you do an ANOVA? What are these statistical evaluation? Or do you do any other statistical evaluation like RSD, SDs? All these are all information that need to be evaluated as a part of the conclusion being drawn from the result. First, the collection of result from the process, collection of data from the testing, collection of data from the in-process that you are doing as a part of your process validation. All these data need to be evaluated mathematically, because the main is to prove that it is constantly producing delivery quality product, and nothing can, you can prove, you can make thousands of words, but mathematically 1 + 1 is always equal to 2. So if you can prove whatever data you are generating mathematically, it means it is true. So now from the process design stage, okay, as there was also a slide in the GMP, I will because it was such an elaborate slide, it makes my job easier, and thankful to Jen sir again. So the thing is, there is a many part of a process of a product design, and process design is one of the key factors. Now, when I say process design, there are certain key terminology now which is being used as a part of the process validation. Those are called critical quality attributes, CQA. So what are these CQA? The CQAs are what your product will be. Now, if I am having a tablet, it should be white in color, it should give a particular action within 30 minutes or 15 minutes of it taking it, and it should be consistent, and it should not impact give any side effect. These are called the quality attribute. So the quality attributes becomes the specification. So assays, dissolution, content uniformity, these are the terminology you may be reading in your books. So these process design, when I say, and then you are not going to make a tablet just by saying that, okay, I want a white tablet. Now white tablet has to be done by making those controls. So what are these? They are called the CPP, critical process control. Now in critical process control, there are thousands of factors. If I can go on, it will take a day to talk on the CPPs and CQAs. It's such a huge subject. So in that, as I say, I will just use raw material, testing methodology, method validations. All this comes under the CPPs. All this critical process, there are many, like warehousing, the environment, the instrument qualification, the preparation of the document, the preparation of sampling methodology, all that will come as a part of the CPPs. So once we know the CQAs, once we know the CPPs, then we must also understand what are our critical process control parameter. Because when should we draw the sample is defined which particular process we consider as critical to give and assure the product of the quality of the product. So now once we know this overall, now once what we have talked about in this small 1-minute session consists of a process design. Is the product design, QBD, whatever the different terminology can be used, but this comes under the QBD, quality by design. What it means is when you design a product, by no matter who works on that product, who works on that process, whoever tests that product will be able to get a consistent, reproducible, and quality product at the end of the steps. That's what is all quality by design. Through commercial production, why it is commercial production? Because these process validation batches can be marketed, okay, which establish scientific evidence that a process is capable of consistently delivering quality product. The only difference between Schedule M and FDA guideline will be that process validation batches cannot be marketed in India because we don't have an exhibit batch concept which FDA have, but process validation batches can be marketed in FDA. Commercial process validation batches can be marketed only on certain conditions, as to be pre-approved by the CDSO. Now coming on to this is a basic what does the FDA basically calls about, say, process design, and then you have to evaluate, you have to do a process qualification. Now there is many students have asked, or many people are still confused about what is the difference between qualification and validation. Absolutely, to be very clear into and very straightforward, absolutely there is no difference. So qualification is a terminology used by the European people and validation is used by the US people. And you know what, money is all that matters at the end of the day. And US have more money, more power. So validation has come to has been known. And so where the confusion? Because we as a country cater to which have a big pharmaceutical industry or the robust pharmaceutical industry, and we cater to both the continent of US and Europe. So this where the confusion generally arises because of the guidelines, and people read one guideline, don't read the other guideline. So it's a very natural. So if anybody asks what is the difference between qualification validation in your interview, please be assured nothing difference. It's the same, and only one is a European concept and one is a US concept. So the process qualification, as already we have said, it consists of the design of the facility, qualification of the equipment, utilities, and then comes the process qualification. So I will be focusing only on the process qualification part and the continuous process verification, which is the next stage after the process validation. So of course, the continuous process validation will have routine monitoring program. We'll talk about a little brief of it because it's also a very vast subject of how to do. So I will consistently and focus only on the process qualification part. Now, when we talk about this process verification or the process validation, once we must, as I already say, the quality by design. So it's what is the definition? A systematic approach to develop begins with a predefined objective and emphasize product and process understanding and process control based on sound scientific knowledge and quality risk management. So now all these definitions, what people don't see generally is the words that has been used to define them. Now, if you see, it's a systematic approach. That means it cannot be random. I don't wake up in the morning and say that, okay, I'll do a quality by design. So you must have a plan. You must have a roadmap. You must have a protocol. So based upon that, you have to define what you are going to do. Just like when you are trying to, will be doing, it's like many of you guys will be doing your masters and bachelors, you will be told by your professors that, okay, we need to do or develop this. So that we need to do, we need to achieve this is a kind of a statement that defines how you are going to approach your research paper in the future. So to approach through development, that begins with predefined objectives. So you have to predefine your objective, what you want to achieve, emphasize product and process understanding. Now, once you have decide what you have to do, now you have to break down into a different structure. That means you have to say what I am going to do at the end of the day. Step 1, step 2, I will collect this data, I will do literature survey. That's what you generally do when you are doing a research paper. So similarly with process validation, you must understand the process. So what happens with the process? Now, if I ask you what is a process, like you driving a vehicle is also a process. Now, when remember whoever who drives here, there are so many students who will be driving. I have a question. Do you still remember that the way you start driving on the first day? Now you will very careful. You are consciously looking at the other traffic that's coming from other direction, and you are driving a little slowly. Now over a period of 15 or 20 days of constant riding, you gain confidence, and you could, your body, your muscles get toned to react to certain things subconscious level. So now that's called process understanding. That means with the overall time, you gain a knowledge about how the process works and how the things are going on. So you become more confident. So similarly, now every day, sometime when it is raining, after suppose you after driven for 15 days, on the 16th day it rains. Now suddenly again you are more careful because you have not driven in rain before. So similarly, these rain are known as variability. So similarly, in process validation, we have called different variability. Now the variability can be from the raw material. When do I say raw material, it means what goes into the process validation? First, people, raw material, process, utilities, all these are variable. Means today you may be getting a material from a source which has an assay of say 99. Tomorrow you may be getting a material which has an assay of say 92. So now these kind of variability has an impact, and many raw materials have different particle sizes. This defines a lot of changes, a lot of things in the process design. So to know how much is allowed, the quality by design give us the boundary, the overall boundary. Okay, this is your land on which you can build your house. The house, say the process validation. Now the process validation decides what is the boundary of what is the house, how big it will be. You can step out of your house, you'll still be within compliance, but if you stay within the house, you'll be more protected. So process quality by design gives you the boundary, so it will be very wide. When you do a process validation, you cut down, you try to control it, that I will not step out of the house, because if I step out of the house, I'll get wet or I'll feel the heat. So similarly, these kind of variability need to be studied at the time of the process validation. That's why the current focus while submitting the exhibit batches, which are kind of a process validation batches, but on a smaller scale for product approval, says that you need to manufacture 3 batches, in which one batch need to be manufactured with 2 different lot of API. This is now the standard practice, and it's also now a guideline which every industry follow. So basically, you need, why this 2 API need to be there in our, or you need to use 2 different batch of API in the batch to ensure that the variability is also covered as a part of the process development. So now once you know what are the variables, you need to control those variables. Like in case of a compression machine, the RPM of the compression machine, the compaction force of the compression machine, or in case of a FBD, that is a fluid bed dryer, the inlet temperature, what is the exhaust temperature, what was the LOD of that batch, those kind of things are called different variables which need to be controlled within a particular range. This is established during the process validation. So what I did, so when we are doing an R&D, say we can say the scientist said we can dry the material from 40 to 60. In the validation, we cut down. No, we will not go till 40. We'll from 45 to say 50 will dry. So what we are doing is that we are decreasing the chance of variability by identifying it. This temperature, this particle size becomes our critical process control parameters. So this need to be defined in a process validation. Now, when you will join the industry, the first thing that any person will ask is the protocol ready? That is, the process validation protocol is ready or not? The process validation protocol is the first document which defines how the process validation methodology will be done. Generally, if I ask any of these people or any of the people that what is the process validation, they will say that we need to manufacture 3 batch. Okay, that's true. That's what FDA, European Union say, but FDA doesn't say. So the FDA says that it is up to you to decide how many batches you need to manufacture in a process validation. It's not 3, it can be 5, it can be 10. It is based upon your convenience and your ability to evaluate the data to assure the quality of the product is done. So when you are satisfied, it is on the emphasis on the industry. The FDA says it is up to the industry people to decide how much validation is enough validation. People generally follow the 3 batch principle, but it is not the sign which everybody follow for every single product. So that is one keynote I just say while we are defining the process variability. Now moving on to the control, already we have talked about control of variability. All these are part of the process validation document. In the process validation document, we decide from which of this critical process or stage we need to take out sample. Now, once we have taken out sample, we are not going to keep it for ourselves. We are going to test it. Now, we are not going to test everything, because if I am going to, if the machine is only drying, then the first thing is what was the water content before and what is the water content after. We don't need to test for assay at that period, because we are not going to see any difference in the assay. May or may not, but we don't need, because the critical point and the fact that must be understand is that that equipment is basically for drying. So if it is drying properly, I am satisfied with the process, because this is the process validation. The final product should come out of this process validation, and that's the reason we don't need to test it for assay at that particular time. So when you are doing a process validation, there is no rule. You have to be scientifically able to evaluate that what the process is doing, what the equipment is delivering, how the granules or the particle size are changing in that machine, and then decide which are the critical attributes of the product which is impacted by the process, impacted by the equipment, and decide your testing criteria. Now, this testing criteria need to be evaluated between a particular specification. You can't have a LOD. So one batch you got 6, one batch you got 8, one batch you got 2. Is it validated? No, dear. It's absolutely not. Your process design may say that up to 6 it is okay. But in that case, you can't say every time if it will give you a 6, it will okay, because that's your boundary. Remember the house example, it's the boundary which, so for the operator to actually practically do it, you need to squeeze it a little bit. Okay, you need to target of LOD of 3%. So even if it 4% comes around 4.5, we'll consider acceptable. So the target will be 3. So once 3 is achieved, we know that even if it increases later on during storage, it will be still within the 6 limit that I have set for myself. But if I put target 6, what happened during storage, if the material is hygroscopic, it will gain those moisture, and we are not testing every day. So what will happen from 6 to 7, it will jump and we'll don't know. So there will be a lot of problem during the compression where we'll see sticking, picking, and other defects on their tablets. So now once we understand the variability, we have understand and design a sampling plan, we need to execute the protocol. So summarizing what I have just said, first understanding the process, what are these, a wet granulation process, is a dry granulation process, all those, and based upon these basic scientific knowledge, we decide to know which equipment we'll be using. So all the equipment must be qualified. That's the basic demand of the process validation before you start. So all your equipment are qualified, all your utilities are qualified or validated, any terminology you like to use. So after that, your raw material are tested and approved by, you have a specification, you have a scope of variability for your raw material, it is tested and approved, and then you decide your process variability. You define those process variability, you define what test you are going to do at different stages of your process, and then you draw a sampling plan. Sampling plan is, are you going to take one sample, you are going to take a triplicate sample, or how the sample will be taken. It is going to be taken as a part of, you are going to use a sampling thief, or you are going to use a simple, just pick it up through a scoop. It can be as simple as anything, that you just take the sample as it is from any of the drum. Or you may have a statistical formula that if it is a big batch, there are 50 container, only I will test only every fifth container. So again, all those sampling methodology or the sampling technique that you need to be justified. When I say justified, it should be scientifically explainable to the auditors, and why the rationale behind choosing every 5th container or 10th container should also be documented as a part of your process validation process. So in industry, this is how we do it. So as I said, I already said during my course of the explanation of those language, process and product knowledge gathered to develop a control strategy. So these control strategy that we develop, or that we put one after another, is compiled into the protocol. So the key function of a process validation is to control the variability. If you know your roads are maintained well and there is no traffic jam, the Google map is right. But if you put a cow in the middle of the road, the Google map doesn't know. So those kind of variability cannot be defined. So there will always be some kind of a cow coming in between the process. The machine breakdown, the utility failed. So how robust is your process, so that even if the machine broke down for 2 hours, how much the process is impacted or not? So those kind of understanding must be there before we can say our process is validated. Now, this is a very new concept which everybody is talking about. Is called risk assessment, which is actually one decision for this kind of risk assessment, because ICS Q9 is totally based upon which this risk assessment is done. Basically, if I have to explain in a short time, which I always give this example. Now, if I take a gun and shoot at you, so the chances of you dying is very high. So the risk statement will be, if I shoot a gun at any person, the chances of killing that person is very high. So that is the risk. The risk is the person will die. Now, if I give you a bulletproof jacket, if I shoot at you, the chances of you getting killed is still there. The severity doesn't decrease. But since now you have a bulletproof jacket, the likelihood of you sustaining a grave injury lessens. So these kind of thing you have to understand. So now the putting a bulletproof jacket is kind of a risk mitigation plan. So similarly in process validation, now we know that say LOD of a hygroscopic material is very critical. It will cause problem in the compression machine. We cannot manufacture a tablet or we cannot fill into a capsule. So what is the control strategy? We need to control the LOD. Now you can control the LOD while it is on the machine where you can test and say, but what about the storage? That's where the risk assessment helps. So what the risk assessment say? It says hygroscopic material, that's the risk. So what is the severity? It will cause sticking in the tablet. So what is the mitigation plan? During the storage of these granules or during the storage of these dried granules, you need to put some silica gel bags in between the two poly bags so that the moisture inside the container are trapped by this silica gel and the product is protected. Until you do this, until you visualize all this risk, now moisture becomes the variable factor here. So similarly, these kind of risk assessment need to be done for each process, each material, each equipment variables. So it's a big document what we are talking about right now. So this risk assessment, you can say that some of the risk will not be so severe. Now, suppose the material is not hygroscopic, but still it may gain little moisture, but little moisture will not impact the process compression. In that case, you don't need to put the silica gel. But when you are taking all this decision, it has to be based upon some scientific rationale, which is documented. You're not taking random that, okay, Sahu said you do this, but Sahu doesn't say without a rationale. You can't follow me by blindly. You have to ask questions. So you have to ask why we need to do that. Why we are doing this? Is there no other way of doing it? Is there not a better way to do it? Is the silica gel the only capable thing? All this activity is documented as a part of the risk assessment process, which is very critical because this risk assessment precautions, risk assessment mitigation plan to be taken are part of your process validation document or the process validation protocol. So efficient planning, because of course you need to do efficient planning if you are going to without training. If you don't train your people on the process validation protocol and you give them the protocol, okay guys, come on now, today we are going to do a process validation, let's execute it. That doesn't work. Learning doesn't come with only studying. Learning also comes with a multiple approach of studying, doing, visualizing, talking, any kind. All this combinedly helps you to gain the knowledge. So that's what, as I was saying, previous experience, some of these batches we may have validated. Now you are increasing the batch size, so you know what you are doing. So that will help. Analytical and product data from clinical studies. This is a part of the exhibit batch and before approval documentation that also helps sometime. Process development information. What I have explained that it's an hygroscopic material. This process needs to be controlled at particular stage are all captured in a document called the product development report. The PDR report is submitted to FDA as part of the approval process. So that is a very valuable document and must be referred at all time to mitigate, to solve issues that might come up later on stage. Many companies do this, which I generally don't recommend if the product has been designed very well, but people are very always cautious. Sometimes the material are very expensive. So before doing a big scale production batch, they want to do, because sometimes the approval process takes about 2 to 3 years sometime. So by the time the company has invested in other technologies or in new machines for which parameter may not be as simple, or the parameters may be more controlled through a PLC based control. When the batch was initially manufactured, it was a mechanical machine. All these variables come up. So during the risk assessment, before the validation, we decide, okay, the exhibit batch was manufactured on a, say on a mechanical machine. Now we have a much better machine after 3 years. Now we don't know the various mechanical parameters which will impact, say, the granule size after sieving. So all those things need to be evaluated, and we don't know data because we don't have any data, because we have not used that machine on that product any longer. So sometimes we take called a scale-up batch. That's a general terminology used. It is called a scale-up batch. Some people call it the pre-validation batches. Terminology doesn't matter. The intent matters. The intent should be to understand the machine or the process, or you to gain some knowledge. Irrespective, many companies don't make any proper documentation on that, but I believe that a report should be prepared for a scale-up report, which I always insist for any organization I have worked with. So, because what did you learn if you don't document? You will lose it. Like, okay, the verbal communication can go on, but if the person dies, the knowledge goes away with the person. So this has happened in history in India before. So it's better to document rather than believe on somebody and say that, okay, we'll do this later. Of course, technology transfer, when I say technology transfer, it is about lot of technology goes into it. Like sometime you may be manufacturing in India, now you want to manufacture back in US. So a lot of that is called a technology transfer from India to US or US to India. When you do that, when all the knowledge that you have acquired over the product need to be transferred, that is the part of the technology transfer document. It includes documentation, it includes the experience, the failures, this includes the previous good batches, all this part of the overall assessment process. This is how a risk assessment basically looks like. So these are the critical quality attributes, CQAs. So these are the parameters which I must achieve as a part of the process validation, is on my left hand side. On the tops, you can see dispensing, bending, granulation, drying. These are the different process steps. And this color coding is done, is that the red ones are generally called as the critical ones because it will impact some of the quality attributes of my products. So those need to be controlled. So those where the red ones are identified, generally we draw some kind of a sample or we record some kind of a parameter in detail to control my process. So important output of stage one is that the process, the basic knowledge, what I want to assure you here is that the basic principle of process validation is to control the process, and it should remain within a control state, and that's what we want to achieve. So the best example of process validation is what I always also explain is the biscuit industry. Now, doesn't matter where the Britannia milk bikis or Marigold is manufactured, the taste remains the same. It's an amazing way of validating the product. Even the taste remains the same. Forget about the sugar content, the sweetness of. Have you ever, any of you have ever seen how the taste of a biscuit never change? Because if it change, then the biscuit being manufactured in Tamil Nadu will not work in West Bengal, or biscuit manufactured in West Bengal will not work in the Rajasthan area. So amazing process validation in food industry. I think the best kind of process validation is being done by food industry. That is my personal opinion. So that is what we want to achieve. Imagine the control. The baking process is controlled so much that they know at what particular time the biscuit need to be taken out. Amazing. So that's what excites. That's what excites. That kind of a knowledge is what I try to achieve. I'm still learning. So all these kind of elements of each unit operation, what the machine speed, what is the machine utility, pressures, how much steam is going into the machine, all these kind of, see, it's not simple process validation. It is unique. Like every person is unique and every person's knowledge and looks are unique. So these are very unique things. It cannot be straitjacket. Yes, there can be some guideline, but it cannot be. You have to remember, as a person, when you will join industry, try to look forward to contribute. You will learn, but at the same time, you can contribute because you have a scientific mind. You may have less knowledge, but you can still contribute. You can ask by asking. How can you contribute? You can contribute by asking scientific based question. Why? Why we are doing this? How we are doing this? Why can't it be done by different way? Go to the machine, sit with the machine. Many of the people I have seen young guys don't go to the machine. You can't learn without going to the machine. Please go to the machine. Sit down on the ground and learn the machine. Try to do a screw. These kind of experiences helps. Okay, so all product attributes and process parameters should be in a control strategy. Like this kind of steps are given. These are generally part of the BMRs, where the blending time has to be 5 minutes at particular rotation. It should be given in a V blender or a octagonal blender or a simple bin blender. Doesn't matter. So these kind of parameter and specification are part of the BMR, where it is said you have to inlet temperature for the first 15 minutes shall be 15 to 30 degrees centigrade only. The next 2 hours it will be say should be increased every 5 minutes by 2 degrees centigrade to achieve 40, and should be dried from 40 to 50 for next 2 hours to achieve a LOD of say 3% wet by volume. So these kind of wet by wet, LOD is never by volume. So these kind of things are part of your BMR process. So define CQAs I have already told all this. Risk assessment, again we have told. Process information, input and output, what is going in, what is coming out, we have covered. Parameters and range, as I say, for 30 minutes 15 to 30 degrees centigrade dry. After that, increase by 1 degree centigrade or 2 degrees centigrade for the next 30 minutes, and then reach the temperature of 40 to 50 and maintain it, and then do that. So these kind of called for parameters and range. Of course, some of the attribute right assays should be between 95 to 100%. Those are also coming under the parameter. Design space information, if applicable. Why? Because I don't want to go there. I want a better control. I don't need to step out of the house. So I want to remain in my house. So all this, when we'll continuously do this, what it will show that we are able to control the process. We are able to control the process validation, achieve a reproducible batches. So remember, a batch cannot be released until the process validation report is signed off and concluded. So first is the process validation, execution, testing, compilation of the data and the report, and then comes the final release of the product only when the quality assurance principle says that okay, it is meeting the criteria. So the number of PB batches to be produced should be justified, as I've already told earlier. Full scale, as I've told you, we sometimes do a small pre-validation batch, and then we do a commercial batch depending upon how costly the materials are. So all the SOPs, all the systems, the controls are part of your process. All these things that you are seeing on the screen are part of the process validation report once the data has been collected. So what has to be, what impacts all this is that, as I've already mentioned, all this I have already talked about it, that raw material is a variable. So you need to sample them beforehand, you need to test them, you need to release them before you can use them in the process validation. Different plants of manufacture, that's a part of the technology transfer that we talked about. Different process equipment for use with the same process, like exhibit batch was on a manual process. Now we are coming back to a PLC batch, much controlled process. So you need to know your equipment. Of course, operator to operator variability will be there. Some operator, as I said about attitude, some operator will be very cautious. They will try to understand the process. Some operator say, okay, fine, as long as the product is meeting the hardness, thickness, I will not worry about what RPM I am running the batch, as long as even if the machine is making some abnormal sound. So coming to the sound, I would like to say a small experience. That's a difference between culture, difference between society. I was thankful I worked in Japan for a few years. One day I was walking there, and people tell me that there was a process validation going on, and I found 8 people surrounding a compression machine, and all of them were standing, like surrounding the machine, and they were just standing there and not talking to each other, just listening. Now I was surprised, because at that time I was about 8-9 years experience, and I never seen like people, I don't know, 8 people in the machine. I thought some problem is going on, so let me find out. As a quality assurance guy, I said that let me find out what the problem is, and I didn't see that machine was coming out, and the tablets were coming out good. And then I asked one of the guy, what are you doing, guys? We are listening to the sound of the machine. Now that was a very cultural shock for me, because no matter wherever you will go or work in any industry, And we never listen to the sound of the machine, because we consider that it's very normal. As long as the machine is giving us good tablet, we don't worry about what is happening to the machine. But the thing is, the process validation depends upon the machine. If the machine is getting wear and tear very quickly, that means it requires regular maintenance after every batch. And that can only be addressed if we know how the machine is behaving on normally, what is the sound level, and how the machine is behaving when we putting that particular batch onto the validation. These kind of documentation, this shows the attitude. That's why what I'm saying, it's not written somewhere that you need to listen to the sound, but to understand the process is your baby. Sometimes a mother knows the baby is hungry. The child is not crying, but she sees the baby's face and knows. That kind of understanding is expected, and this is what I'm trying to learn as a part of my still learning procedure. So different lab facility and analyst capability. Of course, human variations cannot be accounted no matter how much we try, but there will be. But if it can be evaluated based upon statistical analysis, very well done for the process validation report. So just to touch upon this, this is not a part of the thing, but it's a major part of the process validation is the different kind of sampling that can be like these are the different methods. Simple random sampling, you go into the room, pick random sampling from here and there. Otherwise, the stratified random sampling means you divide. So you know your process is going to be say 2 hours long, and your process validation protocol says, okay, you have to take a sample every half an hour, or say you have to take a sample after every 1 kg of a batch. That's called a stratified random sampling. Nested sampling, when after the process is completed, you put it into different lots. You divide that into different lots, like put into a different container for a simple explanation. So from sampling from each bucket or each container becomes a kind of a nested sampling. Systematic sampling, again, people generally confuse with the stratified and random and systematic sampling. Stratified sampling is a specific location or a specific period, while systematic sampling is where you collect a sample throughout the process run. It is not equally like every 10 minutes. No, it can be 10 minutes. Another sampling can be 15 minutes, 20 minutes, and so on. So this kind of thing is called the systematic sampling. So again, just to touch upon the continuous process verification, the continuous process verification will be a part of the monitoring process, where after the validation batches are over, you keep on monitoring the critical process parameter as a part of your batch record, and you compile them into a report at the end of the year called the 𝗵𝗄. And all the change control, the change control is nothing but the changes that you do with your process or your document or your methods. All those will data trending. That's why I say the statistical review of all those data, and different things are compiled into a single report called the 𝗵𝗄, and you match that with the process validation report. That matching of process validation report and your 𝗵𝗄 data is a kind of a continuous process verification. There are other PAT technology which are very advanced in, and is not a part of this presentation right now because it's too advanced for you. So those kind of things are called continuous process verification. So we have already talked about this: control range of critical parameter, control strategy, design space. So all this parameter to control these data are part of this process validation process. So again, all changes, track and trended, plan unplanned changes. Sometimes when we are manufacturing, we may come after a failure. Like suddenly some of the tablets found broken in one of the bucket. These are unplanned. So it may be a specific cause due to a mechanical issue that at particular time an operator did some change in the machine parameter which causes those broken tablet, or it could be a systematic error where over a period of time after manufacturing 50 or 60 batch, the machine has developed some fault and some key machine parts need to be replaced. So this need to be investigated. Each failure is investigated, which is again unique, and we need to understand how quickly, how frequently this is happening based upon the data that we collect. So that's why pharmaceutical industry is a very data oriented process. But the sad part is we collect a lot of data, but we don't evaluate the data. And when we don't evaluate, we don't know what to do. and we just react to a knee-jerk reaction, which sometimes works, sometimes doesn't. And end of the day, we blame some other person or the process to that we are not able to do this and this was not manufactured properly. No, it is must be understand that over a period of time things change and need to be evaluated in a different angle. So demonstrating consistency of initial results, statistical process control, as I already talked about, data from automation, that is a big thing, that all the data what is being collected from the machines or process are collected directly through SCADA systems. There are many different kind of systems which collect data automatically and gives you an evaluation, the graph. Again, we are lazy. Human being by nature is lazy. Now we have SCADA which collects the data, so you don't need to do any work. It even interprets the data. It gives you a graph directly. You don't even need to draw it. But machine cannot be evaluating it. It cannot give you a conclusion. Okay, at the end of the day, it will say, okay, fine, now you change the machine. This machine is not working, go to the next machine. That cannot evaluate. So even human beings are trying to develop that. That's called the artificial intelligence. So we are becoming lazier day by day. So one day it will come that we are so dependent that we'll forget how to use our brain. So please don't stop doing that. Even if there is an artificial intelligence, please use human intelligence. So risk-based monitoring program, of course, that is again a part of the risk, which process continue to be monitored. Okay, these are again trending data, that is a continuous verification process. I have a less time, I have some meetings, so I will skip this part today. So the periodic review has to be there. The APQs need to be prepared, then documented, conclusion should be done, and the improvement plan should be there. This is not this may be the last slide, but what I want to share today is that as a part of the manufacturing industry is that things can be as easy as it is possible and can be as complicated as possible. It is up to us how we see the things. We can have a learning attitude, we can have an appeasement attitude that we deserve because we have some degree. For all newcomers who are listening to me, and the only advice is we have 360 days, 365 days in a calendar year. Make it a point that each day before you go to sleep, you should learn one new thing. It can be technical, it can be non-technical, doesn't matter. And read 3 pages of something meaningful in your life. Like it can be a religious book, it can be a motivational book, it can be self-improvement books, but something which is meaningful. 3 pages in a day, and one learning before you go to sleep, or any knowledge or any fact which you never knew before you go to sleep. You have 365 learning at the end of the year, and you will be a better person at the end of the year, and that should be your first goal. And in industry, people with right attitude and skill sets which can be developed as you learn. So always remember, whenever you join industry, the first thing is you have a learning attitude and you try to develop the skill sets which is required by the industry. That will give you success in the long run. So with this, I end my presentation and I will take questions now. Please feel to ask any question on any subject and I'll be happy to answer right now.
Thank you, sir, for teaching us about the validation process and stages in detail. We are indebted to you for giving your treasure of knowledge. Your session has made the pharmaceutical process validation concept so easy to understand, and it is real experience of learning all this apart from the books. Thank you very much, sir. Now, moving forward, we have question answering session. I request the viewer of the session to please drop your question in the chat box. With your permission, sir, I would like to ask you some questions which have been asked by our audiences. Sir, you told us about something quality by design. So is it a part of validation process?
No, it is not a part of the validation process. It is a prelude to the validation process. Quality by design things are documented separately. That is a part of the process development report and is one of the report which is what you call referred before when you are designing the process validation protocol. So from the process quality by design document, you know what is the limit you can go, extent you can go. Beyond that, FDA will not accept it, because you have already said that to FDA while you are getting your approval that I will stay within this limit. But as a part of the process validation, you want to squeeze and become more tighter so that your process will not fail. So sometimes due to a machine failure, or sometimes due to XYZ or human variations, something went wrong, you will be still within the acceptable range that you have submitted as a part of your process or product design. It's not the process, it's a part of the product design. So it is not a part of the process validation, but it is referred as a part of the process validation.
Thank you, sir. The next question is, how can we practice and learn validation process at institute level, like many other experiments we perform in labs?
Go into the kitchen, walk with your mom. You want to learn about validation? Learn how to make a atta gunte hai na, what you call that? I don't know in English what it is called, but in Hindi it is called, that is a granulation process. That's the granulation process. So if you can make a soft dough for your mother, you know a part of the granulation. And I believe that everybody has a mom. Definitely everybody is born, so everybody has a mom. And have you seen your mother cooking change? The particular curry that they prepared, it tastes the same every single day. Yes. Doesn't matter how bad your mother's mood is, or you had a father and mother had a fight, still the curry tastes the same. That's validation. Go to the kitchen if you want to learn validation at the institute level. Learn what are the things. All mom prepare differently. Some people put the curry patta first into the oil. Some people doesn't. The taste changes. That's all it matters. People know that all these spices need to go with the oil. Why? Because the aromatic, it's an aromatic things that need to come out. Water will not allow it to come out. So, and oil is required. These are process validation. People know, our grandmother, our mother know. The thing is, learn to prepare a dal first. Yes. Learn to prepare a dal. That's validation, man.
Thank you, sir. Sir, what are the common mistakes takes place in process validation and how those can be avoided?
The common process mistakes that I have seen over a period of is that we don't refer all the documents. Like when you will be working in industry, they will be giving you a lot of document. They will give you the PDR, they will give you the risk assessment, they will give you all this blah, 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 blah documents. And suddenly you don't know from what to take what. So before you start working, and people don't read all the document, sometimes they don't understand a simple thing, and which could lead to a very bigger problem that I've seen, that because that while doing a technology transfer from say India to Japan and Japan to India, there are some terminology which have multiple meaning in Japanese. So there was a known case like that. It says sieving, and there is a difference between screening and sieving. So the sieving and screening has the same terminology in Japan. So they end up screening the process and they never achieve the granule size and say that the Indian process is very bad. I said, come on, you didn't understand when I. So this kind of things can happen vice versa. So thing is misunderstanding of the process, not knowing what to look for, not documenting or collecting the data correctly. Like there are people like say attitude. Some in process guy will say, okay, yeah, every day I do the same thing. Why they want to test the hardness? Already I have tested 5 times. It is 6 to 8 only. I will write all 6.2, 6.3, 6.4. No, because when you are doing, you are actually not testing. That means your integrity is not right. You are cheating there. So what happened during that time? The process may be running at 8 to 10, and all your data shows 6 to 8 because that's what you have assumed. It is not the actual data. So thing is, people with the right attitude should be there in the validation, and they should collect the data in a true manner. It can be data which is not acceptable. That's okay, but that helps you to understand. Sometimes when there is a problem in our life, it makes us a better person. Same way with the process validation. When there is a problem, it makes the process better. So the main problem is we don't know what to document. We don't know what to document, and that has led to most of the problem in process validation.
Thank you, sir. The next question is there any difference in process validation of generic and ethical drug manufacturing? If yes, please explain.
No, there is no difference. As I say, principally, there is no difference. Doesn't matter if you manufacture a biscuit, your mother manufacturing a dal, and I am manufacturing a tablet. The process understanding remains the same. Only the terminology thing, छूंक लगाना है या granulation करना है. That's the only terminology difference that I can think about. But principally wise, whatever you will be reading in your books, textbooks are absolutely the same. Be assured.
Thank you, sir. The next question is, if once the drug is manufactured and then we come to know that compound not follow the GMP guidelines, then what happens to the product, or what happens with recalled products?
If it is not manufactured and it has been dispatched, it's a different thing. If it is within the facility, it's a different thing. So if it is within the facility, we control it, we destroy it, we investigate what caused the failure. But if it is already distributed and we came to know at a later stage, it's a little messy affair. Lot of legal requirements are there, which is like, too detailed. You can go on days on day on how to do a recall. It's a, I should not use that language. It's a very big pain, and it's a time consuming thing. It's very bad for a company. It's takes a lot of toll on the manpower and cost a company a lot. So I will rather have a situation where I'm in control. I came to know and it is within my control, rather than have a situation where the batch is already released and we need to recall. And I don't believe in reprocessing any batches. So that's my personal opinion. Many people do. They are right, they are wrong. But as a quality guy, I don't believe in reprocess. Either you get it right at the first time or you don't get it right.